Hello, my name is Carlos, and today I'm going to talk about the paper BERTology Meets Biology, Interpreting Attention in Protein Language Models by Big et al. The paper was published this year, in 2021, and looks to shed light on the biological knowledge that attention mechanisms stored about protein language. This is a great example of the various applications for the transformer and BERT architectures, and it goes beyond NLP tasks and phrases and an alternative language such as proteins. BERT has also been proven to work with DNA and RNA sequences, highlighting the applicability of the model to biological tasks. This work contains an introduction and background, mostly of biological terms necessary to fully understand the paper. Then the methodology highlights the models, architectures, and the most important part of all of this work is the results, which look at the relationship that exists between attention heads and protein language. So, what are proteins and why do we study them? Proteins are molecules present on every living being. They have a wide variety of shapes and functions, and are in charge of all the processes necessary for life. Chances are that if you think of a biological task, whatever you can think of it, there's a protein that can fulfill that task. So you have proteins that are able to recognize bacteria in your immune system. You have proteins able to carry oxygen as hemoglobin. You have proteins in charge of replication of DNA. So there's a huge amount of proteins with different tasks, with different variety. And all of this variety is due to the fact that they are comprised by a sequence of amino acids. So a protein in its basic form is just this, a sequence of amino acids. And amino acids are small molecules that we represent by a letter in the alphabet. So you will have A representing amino acid alanine, you would have D, G, B, L, C, and so on. So you have 20 essential amino acids for life, and you will represent those 20 with a letter in the alphabet. So the order in the sequence of these letters is what determines the structure of a protein, and the structure of the protein itself determines the function of the protein. So you can see how it becomes a language. So the order of the amino acids, so in this case the order of the letters in the sequence, determines the properties of the protein. So you would have the first structure is the sequence of amino acids. The second one is the formation of these beta sheets or alpha helix. This is because of the interaction of hydrogen atoms in one of the amino acids and oxygen atoms in a close amino acid. So, for example, you would have an interaction here between these two that will prompt an alpha helix, or if you have an interaction that it's more close, it would prompt beta sheets. So this is the secondary structure of the protein. Then you have the tertiary structure of the protein, and this one is the really important one because this one is the one that determines how the protein folds, and those folds create what is called as a binding site. So the binding site, and it's easy to see here in this protein, the binding site is the location of the protein that interacts with the molecule, and it's super specific. So here you can see a DNA strand, and here it's the binding site of the protein. So this is all determined by the tertiary structure. And how do you get to the tertiary structure? You go back to this. to the sequence of amino acids. And what happens is that this amino acid will interact with this one. How that happens? Well, each amino acid has a different chemical composition, and some of them, for example, have sulfur atoms, and the interaction, the chemical interaction that this creates, make the protein fold. So if you were to analyze this in 3D, this sequence in 3D, you will see that this amino acid will be in contact with this amino acid, which is farther down the road, which is farther down the sequence. You can think of a sentence, for example, and this word that is at the beginning of the sentence will interact with a word in the end of the sentence. So you can already see how the protein language works and how we can leverage a transformer architecture to understand things about this language. And so here we can see this sequence of amino acids that form the protein. Here we can see all of the 20 essential amino acids and the sequence in which they are arranged will give a protein its properties. And the last thing we need to know is this. This is a substitution matrix, which basically tells us what happens if we were to change an amino acid for another amino acid. So if we were to change this amino acid, for example, which is valine, we change it for histidine, there's a -3 score, which indicates that there will be a huge change in the protein. Blue values indicate no change, red values indicate a big change. We do not want big changes because sometimes those changes just make the protein unviable. So the model this paper chooses to work with is the transformer. So the transformer has been proved to learn useful representations for protein classification in previous works. The transformer, as you well may know, ditches the recurrent neural network architecture and completely focuses on attention mechanisms. Attention mechanisms, as the name suggests, allow the input to interact with itself and discover which part of the input needs more attention. So what basically an attention mechanism does is you can think of a sentence, and for example you can think of the basic sentence, the cat is on the mat. The cat is on the mat. On the mat. And so what attention does is if you tokenize this, so you have token 1, 2, 3, 4, 5, 6, and if you want to predict this token or analyze this token, we know that is a verb that talks about location. So this part is important, especially on, so because on will tell you that we are talking about where something is, so we need to use this is, and cat, because cat will tell you if you need to use is or are, right? So these two, the model knows that in order to predict is, while all of the sentence is important, the most important tokens in the sentence are cat and on, right? Because it's gonna tell you which verb and the form of the verb. So it's a very simple example, but that's how you can wrap your head around attention. And so you would use the input will be the tokens. The tokens will then need to be joined by a positional encoding to tell the model the sequence in which the tokens work. and you will feed that into the model. The transformer model is composed of an encoder and a decoder, and so the encoder itself will be composed by many layers of this architecture, comprised by a multi-head attention and a feed-forward lineal layer. So you would get the inputs here. Here you would get an output, and the output to the encoder will have all the attention information and will be fed into the decoder. In this case, this decoder is used for machine translation, so we'll predict a sequence, and so the decoder will use these encodings with attention to predict the sequence as correctly as the model can. We can see here that attention is calculated as the matrix dot product of queries, keys. Those are scaled by the length of the sequence, and then there's a softmax operation. You would calculate a softmax function on this product and scaling, and then multiply it by the value corresponding to this key. Each input has a set of keys and values. You can think of them as weights, and the query is what we ask the model. So you can think as the query as in some type of database query, and we have the keys and values that are weights associated to an input. This is the self-attention model, and each one of these will be fed into a multi-head attention where you have values, keys, and carries for all of the hidden states. So this is the basic architecture of a transformer. And then we go to BERT. BERT is a variation of the original architecture of the transformer. And the most important part about BERT is that it used bidirectional stacked encoder attention. So as you can see here, BERT, when analyzing a sequence, it not only looks in this way from left to right, it also takes into account the other way. So it's bidirectional. And why is this important? Because sometimes the meaning of this word will depend on the meaning of a word way down the line. And now this makes complete sense to when you are analyzing a protein. Because if you have a sequence, so let's think of this as a sequence of amino acids that will fall and make a protein. Here, this amino acid here, in this position, may be influenced by this position by virtue of how they are folded. So, if our model is able to understand the context of this sequence and know that in this position, this amino acid is influenced by this other amino acid, then we can leverage the BERT architecture to understand proteins. So you will have all the amino acids, you will tokenize them, and what makes the bidirectionality possible is that you use a mask language. What this means is that you will take one of the tokens, one of the amino acids, and you would change it for a mask token. And you would, in quotes, ask the model, okay, what is this mask token? These mask tokens correspond to which amino acid? So here we see that it corresponds to this combination of GP amino acids, and the model, by virtue of analyzing what it's before and what it's after the mask token, will predict correctly the mask token. So the model pays attention to this direction, in this direction, and in the forward direction, and looks back and understands the context and will predict correctly which amino acids are the correct in this for this mask token. So this is you can already see how this is super important when understanding proteins, as we can understand which amino acid is influenced by another amino acid way down the sequence. So the methodology that this paper uses, they use 5 BERT models. 3 of them are normal BERT models, and then they use 2 variations of Albert and XLNet. You can see here the layers of the encoder, of the transformer encoder. Here you can see the heads, the multi-attention heads they use, the number of parameters, the trained dataset, and the number of sequence on the trained dataset on which these models were pre-trained. TAPE, which is the most important out of all of these models, is a model developed in another paper, which basically tries to understand 5 biological tasks, the 2 most important being these 2: secondary structure prediction and contact prediction. Secondary structure prediction and contact prediction, and if you recall, here we saw secondary structure and contact prediction. So secondary structure will determine this order, the beta sheets or the alpha helixes, and the contacts will determine how the protein folds. So that is super important for us to understand. Okay, and so this is the model that is the flagship right now in protein analysis with BERT. The second type of model, the first variation they use is Albert. Albert is a variation of the original BERT architecture, and the 2 main differences are that first, it shares parameters across layers, while BERT learns unique parameters for every layer. So BERT will learn a set of weights for each layer. So if we come back to BERT, it will learn weights for this layer, and it will learn weights for this other layer. So you will have weights 1 and weights 2 and so on for each layer. In Albert, that does not happen. It's a same set of weights for every other layer. What Albert looks for is to be more efficient, more computationally efficient. And so the second difference is that Albert uses factorized embeddings that allow inputs token to be smaller than the size of the hidden state. So you would take the inputs and you will factorize them and make them basically a smaller matrix for the model to process. And finally, they use XLNet, which is a further improvement of BERT. And this XLNet, instead of using a masked language model, what it does is it calculates the probability of a certain token based in all of the permutations of the sequence. So it will analyze, for example, this sentence: the boat was beached on the riverside. And if you want to understand or predict this token boat, what XLNet will do is look for all of the permutations given this order and given this order. So it will look to, if you are looking to predict boat based on was on riverside, it will have was on riverside, on was riverside, and so on. It would look for all the permutation in order to understand this token. And so those are the 5 models they use. And here you have attention analysis. So the attention analysis, which is the most important part of this whole paper. So you can think, for example, of contact being 2 amino acids being in contact. So for this, they would have a function so that if amino acid I and amino acid J are in contact, it would be equal to 1. They would do this for all of the amino acid pairs. So for all of the amino acid pairs. And once they have all of the amino acid pairs, they would then sum this and look for all of the attention. Here we would look for all of the attention heads that are bigger than theta. And once you get all of the attention heads that are bigger than theta, you just divide it by this so that you get the proportion of attention heads that are giving attention to a certain property. To further analyze this, they design probing tasks. So they would design an embedding probe that was supposed to capture the knowledge encoded in the output embeddings, and an attention probe that will capture the knowledge contained in the attention weights. So these probes are classifiers that predict the contact property or the binding site property or whatever property they would be looking for. The data sets in which they fine-tune the model are these. So we can move to the results, which are quite interesting. And so the first result is that attention aligns strongly with contact maps in deepest layers. So what does this mean? Basically, that when you are looking for contact maps, so we go back to our sequence of amino acids. If you are looking for if this amino acid is in contact with this amino acid, there is an attention head that is in charge of this knowledge. And in the tapered model is this attention head, and for each model it varies, but all in all you can see that this knowledge is encoded in the last layers, in the deepest layers for all of the models. And this makes a lot of sense because attention models have been proven to pay attention to the most complex features in the deepest layers. So this model is understanding first how the sequence works, and then it's somehow understanding too how there is a contact, how there is the folding of the protein. This model understands that the protein folds in a way that allows for this amino acid, let's say A, to have contact with this amino acid B. So this is the model is understanding this. They did not feed structural information, and the possible explanation that the paper suggests is that there exists a statistical, because there's a chemical linkage here, there's also a statistical relation between these two amino acids, and so the model learns that statistical relationship, and it can predict that there is contact between these two amino acids. So the model is actually understanding that the protein folds, and then these two amino acids, by virtue of that folding, are gonna be in contact. The second result talks about binding sites. And so if you remember binding sites, we can go back. Here, binding sites are the location in the sequence which interacts with the target molecule. So in case of the DNA polymerase, that the binding site interacts with DNA. In case of hemoglobin, we will interact with the oxygen atoms, and so on. And so here we see that binding sites are learned through all around the model. And why is that? And this has an incredible biological explanation is because binding sites, let's think of a binding site as this point, binding sites need to be conserved throughout the species, throughout evolution. They are conserved because they need to interact with the properties. So these will not change. These parts will be the same. These will have little variation between proteins, between families of proteins, and the model understands that. The model understands that there can be variation in here, but these parts have to be conserved. And so it makes sense that across the model, across the model, they try to conserve these binding sites. So we can see that actually binding sites are conserved all around the model. So the third result is also really interesting and says that attention heads specialize in particular amino acids. And this statistic tells it all. For 16 out of the 20 amino acids, which means for 80% of all amino acids, there exists an attention head that is solely focused on that amino acid, right? So we can have, for example, this arginine and this amino acid. The attention head in this layer will pay attention to all of these amino acids in the sequence. And we can see that it's the same for this amino acid. There's an attention head that focuses just on this amino acid, right? And it varies for distinct amino acids, but we can say that there's parts of the model that specialize on an amino acid, which is quite interesting because it has further implications. We can study those attention heads to understand the role of an amino acid in a sequence. So that is quite interesting, and it shows here. And finally, and the result that wraps all around, here we have a substitution matrix. And this is an actual substitute matrix calculated with these scores. And so we can see, and these are the attention heads for substitution. So we can see that it's quite similar. We can see, for example, here, we can see here, it's quite similar to here. We can see that here, the attention, the model puts, it's quite similar to a substitution matrix calculated outside of the model. And what this means is that the model is understanding that there are some amino acids that have identical properties, and if you change them, the protein, the meaning of the sequence is conserved, and if you change for other amino acids, the meaning completely changed. So the model is understanding that if you change an amino acid for an amino acid that is similar, the meaning of the protein keeps being the same, and there are some amino acids that if you change that, then the meaning of the whole protein is different. So the model is actually understanding protein language, and we can see that attention is consistent with substitution relationships. And so this work ends concluding that transformer language model recovers structural and functional properties of proteins, as we saw, and that attention mechanisms retain knowledge of high structure features in protein language. We see that language models can be leveraged to uncover novel relationships in biology. So the one thing that we can take away from this work is that we can use deep learning architectures and leverage them to work in other fields that we may not have thought about them at first. The transformer was developed for machine translation, and here we see how a variation, how BERT is being used to understand protein language. And this makes complete sense because as we said in the beginning, BERT will understand a token based on all of the context. And this is essential when studying proteins because an amino acid at the beginning of the sentence can change completely the context of that sentence, right? So there is a relationship between amino acids at the beginning of the sentence and way down the sequence. And if we understand that relationship, we can then study protein language. And we see here that the model actually understands contact maps, it understands a structure, it understands 3D structural information just by knowing the sequence, and this is of the uttermost importance when studying biology, and we can see how we can use these state-of-the-art language models to understand novel relationships in biology. Thank you very much.